Hi, so we'll continue the cells. We are talking about lymphocytes in the previous sessions. So we completed the discussion on lymphocytes. We have seen B cells, T cells and NK cells and their features and mechanism. Now we are going to discuss some other cells, and these cells are collectively the cells we are going to discuss in this part are called as mononuclear phagocytes, or these are sometimes you can see we are familiar with the term called phagocytes, right? These are the cells which are capable of phagocytosis, and the most important phagocytic cell that already we have seen while we discussed the phagocytic mechanism is called the macrophage. So here in this part, two cells we are going to see are, one is monocyte and the other one is macrophage, right? So the monocytes are actually the cells which act as a receptor for this macrophage. So the macrophages will be developed from a monocyte, or monocytes will transform into macrophages during their developmental stages. About 10% of the total leukocytes comprise these mononuclear phagocytes, that means monocytes and macrophages. And these two cells originate from a single progenitor. That progenitor is called the granulocyte monocyte progenitor that we have seen while we see that hematopoiesis. Right, there is a progenitor called granulocyte monocyte progenitor. So this single progenitor is responsible for the synthesis of monocyte. Basically, that's what shown there. They basically synthesize a monocyte, that is promonocyte and then mechomonocyte. This mechomonocyte will be released into the blood circulation. After synthesis and development within the bone marrow, they are released into the blood. Now these monocytes will be present in the blood circulation for around 8 hours. That means for some hours these monocytes will remain in the blood, and after that they will migrate. That means after some time within the blood they will migrate into the tissues. They will migrate into the peripheral sites where the activity of macrophages are required, right? Then when they are shifted from they are migrated from the blood into the tissues, they undergo certain transformation, and then these transformed cells, these more mature, more developed cells are what we call the macrophages. Clear? So these are nothing but the same cells. The monocytes are being transformed into a more potent cell called phagocyte while they are shifting their location from the blood into the peripheral tissues. Fine. So how this conversion occurs? This is nothing but already stated. These are more differentiated and more developed cells of monocytes, and they usually locate tissues where there are certain kind of or at the site of inflammation. Then what are the major changes during this transformation? The most important change is there is a change, or there is an enlargement in the size. There is around 5 to 10 fold increase in size of the macrophage during this transformation. Then the complexity, as expected, the complexity also will be increased. The more organelles will be developed, and all organelles become more functional. Then the phagocytic capacity is greatly enhanced. That means even monocytes can also do phagocytosis, but they are not actively phagocytic cells. Right, we know that. And macrophages are the potent phagocytic cells. So this capacity will be greatly enhanced during this process of transformation, and also they begin to synthesize more chemicals like enzymes and the cytotoxic chemicals which are required for the phagocytic activity. That means for the destruction of the target cell. Then definitely there will be an increased rate of metabolism, because we know there is a kind of metabolic burst during the phagocytic reaction, as the cell require more energy, more proteins, and all the activities are in a boosted situation, so that there will be an increased rate of metabolism for the macrophages. So these are the different changes that accompanied along with the transformation of monocytes into the macrophage, right? Then you can look at this image. You can see the clear image of this. This is about a monocyte. There is nucleus and all those things are there. Lysosome, phagosome, all those things are there. But when they are shifted from the monocyte into the macrophage condition, you can see the more developed or more engaged cytoplasm. You can see more complex cytoplasm, then more pseudopodia. That means this is an actively dividing and actively metabolizing and actively phagocytic cell. Okay. Then about phagocytosis, this need not to be explained in detail because already we have discussed about the mechanism of phagocytosis. So here we know that these phagocytic cells can engulf the foreign substances or a bacteria that will be digested with the help of lysosome. Then there will be phagolysosome formation. Then that ultimately will be destroying this bacteria into small fragments. Then these fragments or the broken products are secreted out by a process called exocytosis. And meanwhile, there is a process called antigen presentation that is done through the class 2 MHC molecule. So these are the events that are happening during the phagocytosis. Right. So as already said, these macrophages are potent APCs, or they have the capacity to present antigens after processing through the phagocytic reactions. Right. So they have the class 2 MHC molecule, as we have seen in the image. They have the class 2 MHC molecule as an important receptor. Right. And these receptors are responsible for the presentation of antigens to the especially to the T helper cell. Right. And not only the class 2 MHC, there are other receptors like CR3 and CR4. They are actually responsible, and not only this one, CR3 and CR4, also the CD16, CD32, 64 and 89. All these are important receptors. They enhance phagocytosis by a mechanism called opsonization. See, what is opsonization? Opsonization is the process by which the phagocytic cell, by which the phagocytosis is enhanced. Basically, this is the process. Opsonization is the process by which the phagocytosis is enhanced. The rate of phagocytosis will be enhanced. And how the rate is enhanced? Because we know for a normal phagocytosis, the phagocytic cell and the target cell need to come together, and the membrane has to be fused together. Right. And only then the phagocytosis and the proceeding mechanism will follow. But then there is a connecting link between these two molecules, because we have the target, we have the macrophage, and we have the target bacteria. So suppose there is a connecting link between the bacteria and the phagocytic cell, then the process will be much faster. All right. So here these receptors plays a crucial role in that way. That means the CR3 or CR4 are the complement receptors, and these are the FC receptors for antibodies. All right. So that what happens, just like as we have seen in the ADCC, these antibody molecules will bind onto the surface of this target molecule. That means onto the surface of the bacteria through their FAB portion. Then the FC portion is free, and this FC portion will interact with these listed receptors like the CD16 or CD32. Or similarly, the same will be the reaction for the complement components also. The complement can interact with the target molecule, and they have the receptor. They have the complementary receptor on the surface of the macrophage, so they can act as a link. They can provide a link there. Okay, let's see the representation. See, suppose you have this. Suppose this is the macrophage, right? And this is the, imagine this is the receptor, and now you have the antibody molecule here. This antibody molecule. Now this antibody has interaction with an antigen. Now this antibody interact with an antigen. So this is the antigen. So the interaction of this is macrophage, and this is antigen. So the interaction between the macrophage and antigen is greatly enhanced by this molecule, right? This is an antibody, actually this is an antibody molecule. So this antibody will act as a bridge between the macrophage and the antigen, so that the rate of phagocytosis will be enhanced. And we know that in other situation, in a normal situation, how phagocytosis occurs. This is the phagocytic cell and this is the bacteria, and the bacteria need to come so close to the phagocytic cell, only then the membrane will fuse. The membrane need to fuse together, then only the phagocytosis is possible, right? And the possibilities are less compared to this one, right? Because here there is a connecting link, connecting external molecule to connect the antigen to the phagocytic cells, right? So this mechanism what we call opsonization. Instead of antibody, the complement proteins can also do the similar function. Okay, so that's about this capacity of these macrophages to act as an antigen presenting cell. Okay, then these are certain receptors commonly found on the macrophage and monocytes. You can see a number of receptors are there. CD6, CD13, 14, 16, 64, etc. are the important things, and still many receptors are there. I am not explaining in detail. Then different types of phagocytes or different types of macrophages based on their location, basically. Then we know these are found throughout the body, right? Macrophages are found throughout the body, in tissues and in circulation, and everywhere you can see these macrophages. They are very important cell for the cell mediated mechanism. Then an important category is called wandering macrophages. These are the macrophages found within the circulation, right? These are the macrophages found within the circulation. The explanation is given, that have recently left the blood during inflammation and moved to the site of infection through positive chemotaxis. That means they have left the blood and moving to the site of inflammation. These are on a move, right? These are wandering macrophages. Then the second class is called fixed macrophages. These are the macrophages residing at a specific tissue or organ, right? Then these fixed macrophages are actually part of reticuloendothelial system and are found in lymph nodes, spleen, as a major cell type along with the other lymphocytes. Okay. Then these are located at various organs and are performing various other functions also in addition to the phagocytosis. Then you can have certain names for the fixed macrophages, like Kupffer cells are the name when they are located at liver cells, mesangial if they are seen in kidneys, microglia when they are located at brain, then osteoclast for bones, then alveolar macrophages for the lungs macrophages, then the peritoneal macrophages if the macrophages are found in the gastrointestinal tract, then histiocytes for connective tissues. So these are the list of macrophages, named macrophages with their corresponding location. And what are the major roles of macrophages? This is also important. What are the features a macrophages, or what are the important functions a macrophage perform within a host body? The first and most important thing is phagocytosis itself. And the mechanism is very well explained before, so that I am not moving into that one. This is the process by which the foreign substances are killed, right? Killing of microbes, infected cells and tumor cells by a process called phagocytosis is done by macrophages. The second mechanism, this is also very crucial, that they can act as APCs, antigen presenting cells. They are responsible for the activation of T lymphocytes, especially the T helper cells. So all those T helper cells will be activated with the help of these antigen presenting cells. All right, and we have seen how this interaction occurs and what are the role of MHC and CD receptors and all. So anyway, macrophages are acting as potent antigen presenting cell. Then inflammatory mediation. That means we know for inflammation, phagocytosis is usually a required mechanism. Normally after phagocytosis there will be inflammation. So there will be a lot of chemicals secreted as a mediator of inflammation through phagocytosis. Then cytokines secreted by these macrophages are also very important performers in the immune system. right. These cytokines play very crucial roles in non-specific body defense. That is important. They are usually involved in the non-specific body, that means cell mediated or not cell mediated, innate mechanisms of the body. Then in addition to these listed mechanisms, they also have role in the development of tissues and organs within the body, like brain, blood cells, mammary gland, pancreas and kidneys, right. So in addition to this immunological function, they also play a certain role in the developmental process. Then when you talk about the role of the cytokines, in which functions they are associated are inflammation, fever development, phagocytosis, T cell activation, the neutrophil attraction and activation, then endothelial cell development, capillary development, etc. These are the important functions these cells are associated. Then important cytokines produced by macrophages include the TNF alpha. These are certain most important cytokines listed. Then you have interleukin 1, then interleukin 6, then interleukin 8. So all these are the important cytokines secreted by these macrophages. So that's about these mononuclear phagocytic cells. So I'll brief again. There are two mononuclear phagocytic cells. One is monocyte, the other one is macrophage. Monocytes are these two cells are synthesized from the common granulocyte monocyte progenitor, and these monocytes are secreted first from the bone marrow into the blood, and after some around 8 hours they will migrate into tissues according to the requirement. And when they reach into the tissues, they have undergone certain changes, some transformation occurs, and there is increase in size, there is more active, and metabolically they become more potentially active, and they become phagocytically also more active, right. And these cells will migrate into various tissues. Then they were called in different names, like Kupffer cells in liver, mesangial cells in, and all those names are there already listed, right. And the major functions of the phagocytic cells are phagocytosis, antigen presentation, and the cytokines perform various different functions, and also they perform a role in inflammation. So all those are the important role. Also they have a certain role in the developmental process of tissues and organs, right. Then the cytokines functions are shown here, and also the major cytokines produced, right. So that's about this mononuclear phagocytes, and especially the macrophages, right. Hope you can follow.